Felicity for that fantastic introduction on what you can do when you do have lots of genomic data and a good reference genome. My talk is more going to be towards those people that don't have that luxury yet. Sorry. I'm going to go straight to the point and say that I've slightly changed the topic of my talk, and the title, sorry, that button. The title that would more closely reflect my talk is A Novel Method to Visualize Pairwise Linkage Disequilibrium in Population Genomics Data and to de novo pull out loci from regions of low recombination, and this will be with focus on inversions. So as you know, the inversion is just a part of a chromosome that's flipped 180 degrees. And what's very characteristic about them is that they form this inversion loop in heterokaryotype, which means that if you get a single recombination here, you will get unbalanced gametes and unviable gametes. But you can still have double recombination inside the inversion loop, and you can, of course, have recombination outside the inversions as well. So the net effect is residual recombination, particularly around the inversion breakpoints. And as we already heard, residual recombination allows adaptive differences to accumulate in the face of gene flow, and this creates sort of genomic islands of speciation, and this is a central concept in speciation, and particularly in ecological speciation. So my study system is Anopheles baimai, which is a major malaria vector in Southeast Asia, Thailand, Myanmar, and Northeast India. And this it belongs to a group of species quite closely related, about 1 to 2 million years. And what's interesting about this group is that there's lots of inversions segregating between them and fixed between the different species. And my species, the baimai, is polymorphic for 5 different inversions. And in particular, these 3 inversions can be found in frequencies up to 50% in different populations throughout the distribution range. There's no closely related reference genome, but we've been able to use Anopheles gambi to some extent. Cytological studies are difficult if you don't have the experience. So the aim of this study was really to focus on molecular genetic work using RAD sequencing to study first the evolutionary history of the inversions potentially. And also we want to use this as a way to get around the need of cytological studies. So our approach was a landscape genomic approach to study inversions de novo. And these are just the sampling sites throughout Myanmar and Northeast India. So we've got 79 unique sites. The idea is that we sample a few samples from many locations from a large geographic area. And the method I'll be focused on today is a use of pairwise measures of linkage disequilibrium from RAD loci and social network analysis to find high LD clusters, which presumably correspond to loci from genomic regions of low recombination, for instance, inversion breakpoints. So once we have these clusters, I'm going to spend a lot of some time to try to convince you that these loci actually come from inversion breakpoints and not from other regions of low recombination. So just for RAD sequencing, we used the SPF1 enzyme, 64 individually barcoded samples per library. We did a de novo assembly of these RAD sequences in stacks. After custom filtering, we had 147 samples from the landscape genomics study and about 3,000 loci with less than 20% missing data across any locus or individual. We also have a linkage map cross. It's an F2 intercross with 4 F1 families with 18 to 19 F2s each, so 84 samples in total. And after removing the barcode and restriction site overhang, we had 90 base pair per RAD tag to play around with. So those of you who are not familiar with RAD sequences, the only thing you need to know is that we got about 3,000 loci scattered around the genome, and we have about 90 base pairs for each of these loci. So the idea is that, so this is gene genealogy showing what we expect from an inversion. So essentially, if you have an inversion, it affects only one chromosome, and then it spreads in the population for one reason or another. And if there is complete lack of recombination, you expect this kind of genealogy. And as you get accumulation of different mutations in those different backgrounds, you also get LD. So when the inversion is very new, you can't it won't affect LD in the population. You can only detect it once the inversion is quite old. Also, you need a population where you have at least some, a good portion of the population consists of individuals with either karyotype. The one thing to remember here is that you mostly get double recombination events between these inversion breakpoints, which means that LD is actually preserved between these inversion breakpoints. So the method we used is actually, in principle, quite simple. We used all the pairwise LD measures. So in this case, we used R squared. So we had about 2.5 million pairwise values. And then we sort them, so having the from highest R squared to lowest. And this is just an example showing 5 different locus pairs with these R squared values, so almost 1. And you can see this locus is this vertex, this locus is this vertex, and this connection here is just because it exists in this data set. And then you can continue having 10 of these, and you can already here actually see lots of loci that are involved in several connections that have several LD values above this cutoff. And if you continue, lowering the cutoff value, you can see that more and more edges are included in the network, and you can see these clusters forming. And this is just to compare it with some random networks, just to show you that these clusters are quite unlikely to form just by chance. And if you lower the cutoff enough, everything will, in the end, will be connected. So next, we used this method to pull out different LD clusters, or LDCs. And these are the specific R squared cutoff values that we used to pull them out. So they were essentially pulled out just before they became connected to another cluster. This is the number of loci we ended up using from each cluster. There was some filtering done based on the value of the R square and the number of edges that locus was involved with. So we got 5 LDCs. And next we did discriminant analysis of principal components, specifically for these LDC loci. And this is just a way to find synthetic variables that show differences between groups while minimizing variation within clusters. So it essentially sorts these individuals into different genetically distinct groups. And what's striking here is that you got, for the 4 first LDCs, you got 3 very distinct groups, whereas the 5th then shows something completely different. And also interesting is that for these 4 first ones, 20 to 122 times more variation explained by the first compared to the second axis. So there seems to be something very significant going on here. And if you think about this picture again with the gene genealogy, it wouldn't be too far-fetched to imagine that these individuals belong to this group are individuals that only have these gene copies from this side, and this then would belong to this side. But then you would also expect the group in the middle here to have much higher heterozygosity, because you put together 2 lineages that have accumulated different mutations, and that's exactly what we see as well. So heterozygosity is 5 times higher in group 2 compared to group 1 and 3. Also, if we analyze LD separately for these, for group 1 and group 2, for all these LDC loci, there is pretty much no LD left. So this supports the inversion hypothesis, and more specifically that this group here is then a heterokaryotype which contains 1 gene copy from each of these sites. The one alternative hypothesis could be that it's just a recombination cold spot, and you have had a very strong population subdivision, and then you have a secondary admixture that creates that LD. So that's why I need to show you also some data from the F2 intercross to see if I can convince you that these indeed are from inversions. So using this DAPC method, we looked at the parents for the F2 cross individually for these 4 groups of loci, and also the F1 individuals. We included that in the RAD dataset as well. You can see that for all LDCs except 2, you have both groups present in the parents. So LDC1 is the only one that is consists of that belongs to this only one group. And when you look at do the network analysis exercise again with this data, because it's a F2 intercross, all the individuals are very related. So you remove all the population structure and everything else that can cause LD. What you expect is then all the loci to cluster according to which chromosome they come from. And this is what we see then using LD based on this F2 intercross. And so basically they cluster into the two chromosomes. So Anopheles has two chromosomes on the X and the sex chromosomes. And using this method, we can assign 75% of the loci to these different chromosomes. And if we look at the blast hits against the Anopheles gambier genome, we can see that all these loci go either to the 2R or the 3L chromosome, and all these loci go to the 2L or the 3R chromosome, and all these then go for the X chromosome. And only about 30% of our loci had significant hits in the gambier genome. That's how distantly related they are. But it was still quite useful to do this exercise. And this suggests that there's been switching of chromosomes since the divergence of Anopheles gambier. Also, what's interesting here that the X chromosome actually aligns to something that looks like a chromosome. And we think that this is because the parents actually belong to one group. So there isn't any inversion segregating in this particular family. So remember that if you only get a double recombination, you also get LD between these ones, and that's what pulls together this one into kind of a mess. So we also looked at the LDC loci, where they belong in this particular network. And so the LDC3 and 4 go to this group. LDC2 goes to the X chromosome, and this LDC1 goes to this chromosome. And so what I want to, for the last slide, I want to focus a bit more on this particular chromosome because it only has one LDC. So what we did is that we compared, so in this LDC we had one F1 family that was, where both parents belonged to the same group. And we did the LD network exercise again. And this time also it aligns to something that looks very closely to a chromosome. And if we compare it to a traditional linkage map, you can see that actually they correspond quite well. And in this case, the LDC1 loci go into this position, and that we think correspond to the inversion breakpoints. So the LDNA method seems to make sense, and these results support the inversion hypothesis. In fact, if I try to do this exercise with any other family than this one, it goes into a mess just because you get LD across these ones as well. So I would be happy to hear if you have any alternative explanations that can explain this. For instance, recombination cold spot we would expect around the centromere, and centromere maybe we would expect to be somewhere here. So that doesn't really fit with the data we see here. It feels much better with the inversion hypothesis. So just as a last slide, because we can sort of karyotype these individuals in silico, we can see the geographic distribution of the karyotypes, and this is for LDC2. And if these all individuals belong to this red group, these all blue individuals belong to this one, and then you've got the heterokaryotypes, and actually correlates very closely with that mountain range here. So potentially there's very interesting stuff going on here. So the questions we can ask now is, what is the geographic distribution of karyotypes? Is demographic population history or selection responsible for geographic distribution of these karyotypes? How are all the inversions, and can we say which one is derived? What are the phylogeographic histories of the inversions, and how different are they to neutral loci? So these are questions we already have some answers to, but I don't have time to talk about today. So I just want to end with acknowledgements. My PI, Kathy Walt, and collaborators that did all the hard fieldwork in Southeast Asia, collecting all the mosquitoes, and then also want to thank lots of other people as well. So I'll be happy to take questions.